Unit 12 in our class deals with how information encoded in DNA turns into traits in organisms. The first step of that process is covered in this video. This is video 12.1 on transcription. There's a lot to cover here, so pause the video if you want to take a closer look at these goals. Otherwise, let's press on. Once the structure of DNA was solved by Watson and Crick, the floodgates opened for how DNA worked. Watson and Crick not only described DNA structure, but also explained semi-conservative replication, and this, the central dogma. The central dogma describes how the flow of genetic information goes from DNA to RNA to protein. We've talked about all of these processes before this year. DNA replication is how DNA makes copies of itself. Transcription gets the information of DNA out of the nucleus by making an RNA copy of the information. Translation converts the nucleotide message of RNA into the amino acid sequence of a protein. This video zooms in on the process of transcription. To get a better sense of what's going on, it's critical to understand that DNA is organized into genes. But we've never really defined a gene. Basically, a gene is a DNA sequence that codes for a protein. There are many genes on each chromosome, and genes are usually thousands of nucleotides long. There are specific DNA sequences within each gene that have specific jobs. Now, the main part is the coding sequence that has the information that's needed to make the protein. The coding sequence starts at the transcription start site right here and ends at the termination site. Then there's the promoter, this yellow part here. Think of a promoter as like a switch that turns the gene on or off. Promoters have a characteristic sequence in them called a Tata box that we'll see later. Most of the times, genes also have enhancer sequences. Once the gene is turned on at the promoter, the enhancer sequences are like volume control knobs that can turn the gene up, increasing gene expression, or turn the gene down, keeping the gene at a low level of expression. The process of transcription makes an RNA copy of the nucleotide code of DNA and makes that information more portable, taking the information from the nucleus to a ribosome. RNA and DNA are a little bit different. First of all, most DNA is double-stranded and most RNA is single-stranded. Although both are made out of nucleotides and have a sugar-phosphate backbone, the sugar and bases used are different. DNA uses deoxyribose, RNA uses ribose. For nitrogenous bases, DNA and RNA both use A, C, and G, but RNA uses uracil, U, whereas DNA uses thymine, T. Moreover, there are a whole bunch of specialized types of RNA, each with a different specialized job that we'll get into over the next few videos. mRNA, rRNA, tRNA, siRNA, and snRNA. If you find yourself getting lost in the molecular biology of transcription, ground yourself in these three phases. Transcription can be broken down into initiation, elongation, and termination. Just like the name suggests, initiation is the start of transcription. Think about initiation as just getting everything ready. Initiation involves two types of proteins and two DNA sequences. The proteins involved are transcription factor proteins and the RNA polymerase enzyme. The DNA sequences involved are the promoter DNA and the enhancer DNA. The promoter DNA is a sequence just upstream of the coding region. Looking at DNA sequences, molecular biologists can find promoters by seeing the Tata box with this characteristic DNA sequence. Now, think of the promoter as kind of a starting gate for RNA polymerase. Transcription factor proteins just help RNA polymerase get to the promoter. Enhancer DNA is typically distant from the promoter, here's an enhancer, and distant from the coding region DNA. Enhancer transcription factor proteins bind at the enhancer DNA sites, and help get RNA polymerase to the promoter. This diagram right here shows all of the major players in place. RNA polymerase is bound to the promoter starting gate DNA with the help of many transcription factors in all of these different colors here, as well as an enhancer transcription factor that's bound to the enhancer DNA sequence. I want to reiterate and clarify the role of transcription factors. Transcription factors are proteins that bind the promoter DNA sequence and enhancer DNA sequences. Transcription factors can fall into two basic categories. Activator transcription factors are transcription factors that help get RNA polymerase to the promoter DNA and thus help increase transcription. Repressor transcription factors destabilize RNA polymerase binding to the promoter region DNA and thus decrease transcription. Here's some beautiful depictions of all of those proteins and DNA sequences gathered together for transcription initiation. All of these blue proteins are transcription factors. These green dots are all transcription factors with RNA polymerase stabled on the promoter, and you can see some activator transcription factors bind to enhancer DNA sequences. The next phase of transcription is elongation. Elongation is where RNA polymerase shoots out of the promoter DNA starting gate, zooms along the coding region, and converts the DNA sequence into an RNA sequence. RNA polymerase can unwind and unzip the DNA on its own. Although RNA polymerase needs transcription factors to tell it where to start, RNA polymerase doesn't need primers to get the process started. You can see a transcription bubble here. That's the spot where RNA polymerase is working. And when you zoom in, you can see RNA polymerase is hard at work making this red RNA strand using the DNA as a template. So, just like DNA polymerase, RNA polymerase adds nucleotides to the 3' end of the strand, and just like DNA polymerase, RNA polymerase uses a DNA strand as a template to know what sequence to build. In fact, the DNA strand that's used by RNA polymerase is called the template strand of DNA. The other DNA strand that isn't used by RNA polymerase is called the non-template strand of DNA. Once RNA polymerase has transcribed the whole RNA coding region, then there's termination. RNA polymerase reaches the terminator DNA sequence, and the newly made RNA is released from RNA polymerase. Now, the RNA is called a pre-RNA. RNA polymerase itself also releases from the DNA to go and transcribe another gene, but there are different plans for the pre-RNA. Before the RNA can be used for translation, the pre-RNA must undergo 3 modifications in the nucleus to become a mature mRNA transcript. The 3 modifications are that the pre-RNA must have a cap put on it, must have a tail put on, and must be spliced. First is the 5' G cap. The cap is put on the 5' end of the pre-RNA, and it's called a G cap because the cap is just a long stretch of modified methylated guanine nucleotides. The purpose of the 5' G cap is to help the RNA get through the nuclear pore and out to the ribosome. Second is the poly A tail. The poly A tail is added to the 3' end, the opposite end of the RNA, as the cap, and it's a long tail of adenine nucleotides. The poly A tail has 2 jobs. First, the length of the tail is kind of like a timer for how much longer the RNA can be used. Over time, the tail gets shorter and shorter, and eventually the RNA is degraded. But as long as the tail is there, the RNA won't be degraded and will keep getting transcribed. Also, the poly A tail is the site of ribosome binding for translation. The third and final modification of pre-RNA is splicing. Splicing is where regions that aren't needed called introns, are taken out of the pre-RNA. Keep in mind that these three modifications only happen in eukaryotic cells, but not in prokaryotic cells like bacteria. I want to add some depth to splicing, what splicing is, how splicing is done, and why splicing is done. The coding region of a gene has instructions to make the protein, but the protein instructions are only in the exon regions of DNA. The intron regions of DNA don't contribute to making the final protein. Now, there's a lot of controversy as to the purpose of introns. Sometimes, introns help make different versions of the same protein, and some scientists think that a lot of times introns are just like junk DNA. But really, I think introns aren't fully understood yet. The key point is here, though, that the introns need to come out, and that's what splicing does. Takes the introns out. Just like everything else in transcription, splicing happens in the nucleus. Here's the nuclear membrane. Think of splicing as cut and paste. The introns are cut out, and the exons are pasted together. After splicing, the pre-RNA is now considered a mature RNA, and the mature RNA leaves the nucleus through the nuclear pores to go get translated. Splicing is done by specialized complex called SNRPs, or SNRMPs, small nuclear ribonucleic particles. Together with the RNA transcript, SNRPs loop the introns to form this structure called a spliceosome before cutting the intron out and gluing the exons back together. One cool thing about having splicing is that cells can use one gene to make many different proteins. Part A up here shows a pre-RNA with all of the introns. But depending on how the introns are spliced, you can make all of these different combinations that are shown in Part B. Overall, the resulting proteins would all have similar jobs, but having these different versions can create specialized proteins for specialized situations. So what did we learn in this video? Let's recap. Genes are DNA regions that encode proteins and have some other specialized DNA sequences as well. Transcription and translation convert the information from genes into a final protein. This video focused on transcription. Transcription initiation involves transcription factor proteins binding to promoter DNA and enhancer DNA to help recruit the big builder enzyme, RNA polymerase, to make RNA. What's made by RNA polymerase is the pre-RNA, and that pre-RNA must undergo 3 post-transcriptional modifications inside of the nucleus before the mRNA becomes a mature RNA that can be translated on ribosomes into proteins.